Hi, my name is Luka Civa and I'm a bachelor student in biology at the University of Copenhagen. And other than studying, I have been working as a research consultant in the Social and Symbiotic Evolution Group led by Professor Michael Poulsen. And here I've been focusing on working with the cultivation of microbes through different research projects. So for my bachelor thesis, I have been investigating a culturomics comparison of microbial diversity across compartments in a Portuguese karst cave ecosystem. I was given a set of sediment samples from a Portuguese cave that is part of a larger interdisciplinary research project named the Barrocal Cave Project, led by Professor Sofia Reboleira.
My name is Kasun Bodawatta and I'm an evolutionary and a community ecologist. My main research area is try to understand ecological drivers influencing microbial community assemblies. And when we started working on those cave sediment samples, we realized, especially when I was doing the literature search, that not many people actually look into these communities, and it's actually very understudied environment. What we did was we decided, okay, we would like to see the spatial structure of the microbial communities in the caves. So then we will sample multiple different regions across the cave along a depth gradient, and also to include other microbes, because our previous work we only look at the bacteria, but then this time we want to look at, along with bacteria, fungal communities and the archaeal communities. Try to get a bit more holistic understanding of the microbial communities in this cave.
Hello, my name is Kajsa Roslund and I'm a postdoc here at University of Copenhagen Center for Volatile Interactions. A group of scientists in Portugal, together with the local government, are establishing a long-term ecological research site in this cave to monitor and preserve the rare ecosystem. There's just one problem. It's difficult to breathe in the cave. The scientists have noticed that during the past 10 years it's getting harder and harder to enter, especially the deepest parts of the cave, because they start to feel lightheaded. And this is where me and our Center for Volatile Interactions, comes in. So I want to investigate where the oxygen is going and what is potentially replacing it. I also want to know what is behind this change in the microclimate of the cave and what could be the potential effects on the whole ecosystem within the cave.
We sampled around 7 locations within the cave. Sediment samples we take, and sometimes in the caves you don't actually have a lot of sediment, so you need to scrape out a little bit from crevices and places along the walls of the cave. But we managed to get enough samples from the 7 locations in the cave, and also we collected samples from outside as a reference sampling point.
Gas samples from different parts of the cave and also outside, and I brought them back to Copenhagen and analyzed them.
So from these samples, I conducted my own isolation experiment with different media and dilution levels across different locations related to the cave. The purpose of this was to try and capture the cultural microbial diversity of this cave with the use of culturomics by counting colony-forming units and categorizing them into different morphotypes, which mean that I differentiated the colony-forming units by macroscopic features such as shape, size, color, and texture. So after incubation and the end of the experiment, a lot of different microbes had appeared. So over the next following months, I managed to isolate over 170 bacterial and fungal isolates with 67 different morphotypes. So based on this, I selected a subset of isolates for DNA sequencing, which revealed a diverse range of fungal genera and a few genera for bacteria. To visually capture the microbes, I've been using the Reshape biotech imaging system from colleagues working with molecular biology over at the Biocenter. And with time-lapse video, it's amazing to see how different they are from each other in terms of morphology and growth rates.
With these extracted DNA samples, we were able to amplify them, these samples for bacteria and fungi and also archaea. And now we are at the process of sequencing them, hoping that we will get the sequencing results soon so we can actually see what are the communities and whether these microbial communities change in the cave depth gradient and also whether it changes across time. Yeah, so now with these samples we are really excited to analyze them, because with this we can get a very good insight into how dynamic the microbial communities within subterranean environment. Not just the spatial structure, we have the temporal structure that also gives an idea how resilient these microbial communities to environmental change or even the seasonal fluctuations in the outside environment. So with this we are hoping we can get a very good characterization of the microbial diversities, and hopefully it will lead to future projects that we can look a bit more into the functional diversity of these microbial communities, and also to understand what proportion of the biological functioning happening within these cave ecosystems are contributed by these tiny microbial communities.
The plan is to look for differences between the gases that are found within the cave and outside, and also in different parts of the cave. The plan is to do another trip to this same cave in November, when I will repeat the measurements. And then the next steps include connecting the gas measurements to other measurements done in the cave, and try to figure out who is producing what and why.
So I am very excited about the future of this project, as we are at the moment making the last volatile organic compound measurements of the same subset of isolates from my thesis together with Kajsa Roslund from Volt. And finally, I hope that this project can be part of something greater, and that it can demonstrate the importance of protecting ecosystems such as caves, because I really think that they have some secret wonders kept in the dark that we don't know of yet.